I spent quite a lot of time debating what to talk about today, so it might be a little bit different than my abstract. I'm conscious that many of you have slightly different backgrounds. So for today, or this morning, should I say, what I'm going to concentrate on is really thinking about how over the last, or increasingly, the mathematical modelling that we do in cancer is becoming much closer to the data, and to sort of talk you through a few case studies that sort of illustrate how availability of better data is the impact that that's having on the sort of modelling work that we're doing. So I guess this is sort of preaching to the converted. We all know that tumours are complicated. We know that there is heterogeneity at many different scales, whether that be at a patient level, whether we look within an individual in terms of their temporal sort of evolution of disease. If we look at a spatial level, tumours themselves are heterogeneous. And that's across many different space and time scales. So it's complicated. So I guess in our modelling work, we operate across all of these different scales. And again, walking around the posters, it's lovely to see the breadth of work that you all are doing, which I think taps into all of these different spatial and temporal scales. So there's lots of excellent, I think, research happening in each of these areas. I guess what I want to do today will also pick up on some work we've been doing that sort of looks at data that can be generated, again, at each of these different spatial and temporal scales of resolution. And I guess, and again, this is preaching to the converted. So given all of that terrible complexity, how can mathematics really do anything that's at all helpful? And again, I think you all know this, but some of the ways that I think we can help is by providing mechanistic insight, where, say, clinicians, experimentalists have hypotheses about how they think a system behaves. We can sort of use our mathematical models to try and test the consistency of those hypotheses, and in that way, I think, provide valuable mechanistic insight. Not only that, I guess we can push our models into new sort of uncharted territory. Once we have a model that's been validated in some sense, we can use our model to generate testable predictions. And again, as a mathematician, one of the things I think that's also especially exciting about working in oncology in particular, but more generally, I think, across biology, is I think the very art, or the very process of trying to develop models of biological systems also throws up new mathematics. Many of the questions we want to address don't fit nicely into existing mathematical frameworks, and I think that's also very stimulating and challenging, depending on your perspective. So I guess that's more just by way of an introduction. And I think the theme that perhaps over sort of underpins what I'll talk about in this first session is thinking about how the wider availability of data, how the impact that that's having on the research that we do in mathematical oncology. And I guess if you've been around for a few years, as I have, you can sort of see how mathematical biology has changed. So when I started doing mathematical biology, we didn't have very good data. If we did, we would have, say, one experiment that was done, one set of measurements in a mouse, something else in some, maybe in vitro, and it was quite a sort of mixed sort of setting. And a lot of the work that we did was really much more fundamental. So we would write down a model, we would solve the equations, and then we would stop. It would be very difficult to fit the model to data. We were just interested very much in qualitative understanding. And then you start to see the rise of fields like computational biology, reflecting, I think, the increasing availability of more data and more detailed data as the technologies advances and computers get better at being able to solve different sorts of equations. Now I think we're moving very much into the era of machine learning, artificial intelligence, et cetera, and I think all of this is reflected in the sort of research that's being done within the community and I guess mathematical biology or computational biology more generally. With all of that as a sort of backdrop, I'm just going to talk about a few very simple, I think, models, but that illustrate in a simple way how we can integrate experimental work and the data that generates with our mathematical models and the sorts of insight that that can provide to us. So this first piece of work is by an ex-PhD student, Marcin, who was adamant that he wanted to go into the lab. He wanted to do experiments and modeling. So these experiments were predominantly experiments that he did. And the questions that we were trying to answer was, how does cellular heterogeneity affect tumor growth? And how does it affect if you have multiple cell lines or different cell populations? How does radiotherapy affect the way in which those cell populations interact? So what Marcin did was he took two different cell lines, either PC3 or DU145, doesn't really matter. I think these are both prostate cancer. And in each case, we had what was considered to be a wild type population, and we had a radiotherapy resistant population. And he grew spheroids from these two wild type and radio resistant populations, either on their own, so a homogeneous tumor spheroid, or he co-cultured them in different sort of combinations, whether that be 50-50, 90-10, etc., etc. And so you can see some of the time courses here. So this is a control, day 3, day 12, etc., and you can see here plotted the growth dynamics of these spheroids over time, and you can notice that there's sort of quantitative and differences in the growth dynamics between the two populations. And here's similar results for the second cell line that he used. Again, control sort of purportedly radio resistant, and this is our radio resistant. And you can see how the time courses change over time. And then additionally what we did was we took these spheroids, we irradiated them, it generated similar data. And then the question we wanted to answer was, first of all, can we predict how the radio-resistant and wild-type populations interact with each other? And do those interactions change following exposure to radiotherapy? And I guess here, is just showing you the differences in the growth dynamics. Here's our control and resistant grown without treatment. Here's following exposure to 6 grays of radiotherapy, and again for the second cell line. So I guess you could say that the resistant, well, you can see perhaps slight differences in these growth dynamics. Maybe the data shows us better. I should say we also performed these experiments using the mixtures of 50-50, etc., etc. And so the idea was to try and predict the growth dynamics of each cell population, and also to try and estimate what the interactions were between the two populations and how those change after a single dose of 6 grays of radiotherapy. Oops, let's... Okay, so the mathematical model that we used is very, very simple. So hopefully everyone is familiar with the Lotka-Volterra equation. So we have our control population denoted by VC. We have our radio-resistant therapy cell line denoted by VR. If we culture them on their own, then we just have logistic growth. And then we have these competition terms, and this is, I guess, what we're really interested in. Lambda R is the impact of the radio-resistant on the control type, and lambda C is the parameter in summarizing the influence of the controls on the radio-resistant. And so what we did was, if we take our pure culture, pure control, pure resistant, then we can fit our logistic growth model to the growth curves, and we can estimate the growth rate R and the carrying capacity for the two cell lines, and you can see the results for the two cell lines here. Okay? And so then the question was, or what we thought would be pretty straightforward, is now we do the same thing. We can hold these parameters fixed and try and estimate lambda R and lambda C from the co-culture experiments. Unfortunately, when we did that, it didn't work. We had no confidence. Our estimates of lambda R and lambda C were they weren't identifiable. And indeed, if you investigate further, then you can see that we can't actually estimate these parameters with any degree of accuracy if we have tumor volume data. So I guess what I should say is that when we do the co-culture, what we're really measuring is VC plus VR. So if we measure the total population, then we can't distinguish accurately lambda R and lambda C. And so that was quite annoying, because these experiments were quite time consuming to do. But then what we did was we decided to see, well, if we can't estimate the parameters from total volumes, what data would we need in order to be able to address them? And I guess you can straightforward to see that if we knew what each population was on its own, then we would be able to estimate what lambda R and lambda C is. But doing that in an experiment is quite difficult to do, because you have to disaggregate your spheroid and you have to separate the two cell lines. And when we sort of mentioned doing this to the experimentalists, they weren't really very pleased with us. They didn't really want to do that, and they certainly didn't want to do it at every time point. And so what we did was we used our mathematical model to generate synthetic data to work out what would be the smallest number of times at which we would need to disaggregate the spheroids, count the proportion of control and resistant, in order to hopefully get accurate estimates of these interaction parameters. And when we did that, we found that a reasonable number would be only 2 or 3, which, given that they were measuring spheroids at, if you can see, we were measuring them fairly often. There's probably about 7 or so time points. So 2 or 3 time points for disaggregating was a lot more reasonable with the experimentalists than they thought we were going to ask them to do it at every single time point. Yes.
On the first slide, very first slide, where you show those, So in the top slide, I see the resistant and the 1:1 control resistant.
Yes.
Those essentially, there's no difference, right?
So yes. So what's happening?
So then why do we even expect any kind of interactions?
Because we start off with a 50-50 mixture. And so what we're saying, what you can see, so good question, it looks like the resistant are overpowering the control, but we want to estimate the parameters. In the second one, that it is dominant, but we're not quite clear what the interactions were.
No, in the first one, the blue and the black, shouldn't they be separate if there is any interaction?
So,
the blue line and the black line in the
so the initial volume is the same. So they start off the same, but you've got half as many of control and half as many of resistance. So what you're seeing there is over time, so the black spots are 100% resistant. The blue diamonds is I start with a 50-50 mixture, so I've got fewer resistant to begin with. So what you're seeing happening, or what you would predict, is that the resistant are winning out. And bear in mind, this is in the absence of treatment, right? There's been no radiotherapy. So what this is saying is that the interaction is that the resistant ones are dominating the control. But that wouldn't be necessarily obvious just because they're radio resistant when you haven't put radiotherapy onto the scene, right? So the logic is right, but still there's a question to try and sort of tease that out. So does that make sense? Yeah, please ask questions right the way through. I'd rather do less. Yeah.
So is there a possibility that factors secreted by the resistant cells is also inducing increased proliferation of the control?
Absolutely, absolutely. So this is, we've simplified, there's lots and lots of possible explanations. So this is a really crude model and designed to be at the level of the data. So if I can't infer the interactions, if I can't fit Lotka-Volterra, doing anything more sophisticated, I'm certainly not going to be able to estimate the parameters. So the idea here is to really have a simple model that's at the level of the data. Then I think if we can fit that model, if we can then look at the nature of the interactions between the two different cell lines, our PC3 and DU145, then we can start to think what's driving those interactions. And indeed, when we look at those results, that did indeed stimulate the experimentalists to do further work, as I will explain to you. I should say, please do ask questions, because I'd rather that if you have got questions, yes, go for it. So I guess what's happened is that the cell lines have been exposed to radiotherapy, so they've kept culturing them, and the ones that survive are the ones that, so they take the basic cell line, irradiate it, keep the ones that survive, keep irradiating it until they get ones that are radio resistant. So they've generated them separately, right? Well, where this seems to contradict that. Yes. Yeah, and I guess, so I don't know whether the experiments you're talking about, I guess there's a difference in culturing things in 2D and in 3D as well, the way that cells might interact with each other. So yes, you make a good point. And to be honest with you, I don't know, I can't remember, or I don't know precisely how these resistant ones were generated. They may have been on a monolayer, which may be different. I don't know, can't remember. So sorry, I hope that clarifies things for you. So, and apologies for not making that clearer as we went through. So where have we got up to? So what we discovered, and I guess this is the point of where the maths and using the maths can be helpful, is if we just measure the total volume, which is our control and resistant, then we can't accurately estimate these interactions, which is important to us. However, we can use our model to generate synthetic data for experimental design, and what we predict is if you measure the proportions at 2 or 3 time points, then you might have a hope of estimating these parameters. And so we convinced the experimentalists that this was worth trying. And so what we found, the spheroids was dissociated at, I think, 3 different time points, and that enabled us to measure the numbers of resistant and control over time, okay, for these 2 different cell lines. So given that data, then we can sort of fit our Lotka-Volterra model, and this is what the data, and I guess just look perhaps at the top line. So this is our PC3 cell line, no radiotherapy. The green is our controls, and then the red, I think, is the resistant, and then we can see the total. And you can see that our model fits. I mean, the data's not fantastic, but I hope you will agree that we can actually fit our model to these data. This is the same cell line, but following exposure to radiotherapy, and you can see there's a slight change in the growth dynamics. Once you dissociate, so when we do the measurements of the proportions, we have to dissociate, but we can use the continued experiments if we want to measure the total volume. Yeah. So we have total volume at finer grain temporal resolution. We have proportions just at 3 time points, but that's sufficient in order to fit the model to the data. And given that that's the painful thing to do, that's quite pleasing. And I guess that's one of the insights that the model gives to us. And so we actually estimated the parameters. So if we get to see what we learnt from this experience, then for one of the cell lines, for the PC3s, what we found was the values of lambda C and lambda R were both positive. So that suggests that there was an antagonistic, and both of them were competing with each other for space. But the result that was surprising to us was that for the second cell line, we found that lambda C was negative, and lambda R, so that the controls were having a positive effect on the resistance. The resistance were antagonistic to the controls. And this was a total surprise to the experimentalists, because we would have thought that everyone would be just competing with each other. So as it turned out, so in the PC3s, we have direct competition between the control and the radio resistant. But for the other cell type, we have this cross reaction where we have an antagonistic relationship between our wild type and resistant cell lines. And as I said, they were eventually very pleased that we did this, because this then stimulated a whole line of further research to try, like you were saying, why are these two cell types interacting with each other differently? And indeed, what they did was they then did fluorescent staining, because there's a big question here. What we can't do is see inside the spheroids. We don't know where the two cell lines are. Is one being pushed out to the center, or is one sort of preferentially in the nutrient-rich region around the outside? Are they mixed together? We really don't know at this stage. The model isn't good enough, the data isn't good enough to give us that insight. But certainly what this can do is throw up some sort of unexpected findings, which then promoted further research where they did actually grow spheroids, as I said, to see where the two different cell lines, how they were localized within the spheroids. But that's, I think, another story. So really, just to summarize this part, what we were trying to do is understand how two different cell lines here, control and resistant populations, interact. And we found in one case we had a direct competitive interaction, but in the second case it was surprisingly an antagonistic relationship. Those interactions didn't change when we added a single dose of radiotherapy, but the magnitudes of the parameters did change. And so I guess the point of this case study is really just to show you how the mathematical modeling aided the design of the experiments. Yes? Mm-hmm.
So in the next round of irradiation, do you see a greater drop in the population?
So we just did a single round of radiotherapy, that was it. But it would be interesting. I mean, there's loads of extra things that you could do with this sort of study, I think, to continue. But to be fair, he had his thesis to do, to write. But lovely question. And again, other things like what's happening, and indeed, I think this is some of the stuff that our experimentalists did to try and understand what growth factors are being secreted by the different cell lines, how are they communicating with each other to give rise to these sorts of interactions. All very, very interesting questions. And again, certainly for the DU145, they would never have thought of doing that if we hadn't sort of encouraged them to do this work. Yeah. Yes, so we fitted it for both. So when we have pure culture, so just a homogeneous, pure control, pure resistant, we estimated the carrying capacities for both and the growth rate. So we did all of that. There are questions you can ask about how you estimate the parameters. Should we pool the data? Should we do it sequentially? Which is how we did it here. And indeed, in further work that's published, I think in Cho et al. So sorry, yeah, this is follow-up work where what we did here, sort of motivated by this study, we developed a cellular automaton model where we took two different cell lines, as in here, and looked at their growth dynamics where we had a known ground truth, and we engineered them to have different characteristics and fitted the model to see the extent to which you can infer what you can infer about the populations when you know what the ground truth is. So if you're interested, and also the questions about how you fit your data to your model, whether you do a sequential thing or whether you pool it. I mean, there's lots of questions that you could ask about that. Yes. I guess you define I will show you on the next few slides. I think, if it is the way that I think it is. So the next bit, moving on, is going to be, we'll be back to what we hope are homogeneous tumor spheroids, and we're going to be looking at some clinical data from a trial at the Moffitt Cancer Institute. And this is really about, so before we were in the lab with tumor spheroids, now we're dealing with clinical data. And this is work by Tom Lewin, who was a PhD student, and Alex Browning, who's a postdoc in Oxford. So this is what I mean by the carrying capacity. So with my tumor spheroid, if I culture it in the lab, eventually it will plateau at a sort of fixed size. So this curve here is showing you data from tumor spheroids, and you can see that over time, the size of the spheroid saturates. And that would be, I guess, when there's a balance between the rate at which cells on the outer edge of the spheroid are proliferating and the rate at which cells towards the center, which are starved of nutrients, the rate at which they're dying. So when those balance, then the overall growth rate will stop, and then you reach what we call a carrying capacity. Does that make sense? Brill. So we've already seen, so this is logistic growth. We've got our growth rate and our carrying capacity. Now, suppose we want to irradiate this tumor, this logistically growing tumor, then the way that we can model that is using, I guess, the linear quadratic model, which is a phenomenological model that's used to predict the cell kill induced by exposure to a dose of radiotherapy, a dose D of radiotherapy. And I guess the usual way in which this is stated is we report a survival fraction. So that's the proportion of cells that remain following exposure to a dose of radiotherapy D, and the parameters alpha and beta are really characterizing the tissue that's being irradiated. And you can see in the plot just above how different values of alpha and beta will give you different sort of plots, different survival fractions for a particular type of tissue, and I guess here different types of radiotherapy doesn't really matter. So if we want to build the simplest model, say, of tumor growth and response to radiotherapy, then what we can do is just combine these two models together. And what we say, so when we're not irradiating, my tumor's growing logistically, I irradiate at a certain time t, and then what I see is I kill off, using my linear quadratic model, a proportion of the tumor cells, and I'm left with a fraction. So if I irradiate at time t, just before time t, my volume is V of t minus, determined from logistic growth. Just after radiotherapy, assuming that the cells are killed instantaneously, magically disappear, then my new volume is going to be the original one multiplied by the survival fraction determined from the linear quadratic growth law. I hope that all makes sense. So just to give you an idea of what that might look like, here are two synthetic tumors with different carrying capacities and different growth rates. So to mimic a patient or what might happen in the clinic, these two tumors grow, then we come to the clinic, or this is our start of treatment. We irradiate Monday through Friday. And so what you see, if I irradiate at T equals 0, my tumour shrinks according to the cell kill induced by radiotherapy. Then it grows the rest of the day. I come back on Tuesday, it shrinks. And so we see this characteristic sawtooth dynamics where we see a drop on exposure when we give our radiotherapy, then regrowth, a drop, etc. And over time, if the treatment is successful, if the cell kill induced by radiotherapy exceeds the growth rate over the sort of period between treatments, then we'll see a gradual reduction in our tumour burden, as you can see here, okay? It's Monday through Friday, weekend off, and I guess five rounds of radiotherapy. A fairly standard, I think, protocol.
So the x-axis is days since treatment.
Yes, so we start treatment at T equals 0. So this is the sort of pre-treatment growth of my two tumours. Does that make sense? Yes. Good question. I will show you. Or I will show you from a particular trial.
One more question. So this model doesn't include any cell cycle arrest.
This is a totally simple model, right? So because think about it, right, what sort of data do you get from a clinical trial? You might get a pre-treatment scan, you might get a measurement at the start of treatment, and then you get a measurement at the end of treatment, 3 time points. And so to do anything very sophisticated, it's a bit like with the other model. You don't have the data, and it's not sufficiently time resolved. So, in fact, one of the unique things about this clinical trial, notice that they actually measured much more often than you would normally do. Typically, you would have pre-treatment, start of treatment, you might have a pre-treatment here, start of treatment, end of treatment. And that's what you're making your judgment about success, failure, etc., right? So this trial was quite unusual, even though it's still only volumes. And to my mind, trying to find out where the hell the tumour is in these, I find quite difficult. Apparently it's here, but to an expert eye, they can see that. So this is kind of quite crude stuff to my mind, but the temporal resolution here is quite unusual and gives us a real hope of trying to fit some sort of mathematical model to it, even though it is quite crude. So this is the real world, and this is a very sort of nice real world. So to answer your question, so this patient, you can imagine if you had filled in the sawtooths in between, might conform to the model that we had on the previous slide, but these others do not. You can see sort of in this case, the patient sort of, they grow, it's a pretty poor, not much happening there. We can see other situations in which the tumour continues to grow for a time and you think, that doesn't look very good, but then they start to respond. So all sorts of things can happen. In this case, they respond, but then they seem to plateau off a bit. So all of these things can happen, but that simple model that I showed will not be able to produce all of these different growth dynamics. Yeah. Yeah. So this is just data from a patient. So these are typical images that we use to generate, to determine what the tumour volumes were. So you don't have information on these subpopulations? This is the sort of information that's available. So it's very crude, right? We don't know, I mean, and we're talking in vivo now. There's blood vessels, there's immune cells, there's loads of things going on. But this is what we know, right? So just tumour volumes, and those tumour volumes, I would say, are a bit approximate, right? Yes. So yes, so I should say I've been slightly, I show these pictures just because I think they're very, very clear. So the measurements are only going up to here. These are predictions of you'll see this picture again. So the measurements are just going up to there. I just didn't cut it off, right? So this is, the dots is what the data is. Here it's so the important thing, I guess, about this, from making the point that not all patients are the same, is that it initially doesn't look as if it's responding, and then it starts to respond, a delayed response, if you like, right? So I guess then the question is, this is the data. The previous logistic growth model, which would be possibly your first go-to, isn't good enough. What can we do? What's the simplest thing we can do? So what we can do is propose a new model, but what should that new model look like, and what's the hypothesis? What are you basing it on? And so the idea that we had, which is quite simple, was, if you think about it, when you irradiate with the logistic model, we assumed that that dead material just was magically removed instantaneously. So you irradiate, you kill cells, and that volume just magically goes away. And so what we want to assume here is it doesn't magically go away straight away. It takes a little bit of time, either for the material to degrade, for immune cells to come in, to phagocytose, to eat it up and clear it, right? So that's what our new model is going to look like. What does it look like? So it's just 2 ODEs. And so here's my viable cells, if you like. They undergo logistic growth. There's a certain rate of, say, necrotic cell death. When the cells die, then that necrotic material or dead cells are removed, if you like, or you can just think of this as natural cell death, doesn't really matter. And then when I irradiate, and I've just changed this ever so slightly from the linear quadratic, basically what this means is I irradiate at a series of time points TI, and when I irradiate, I remove a certain proportion of the tumor volume, gamma. So if you like, this is 1 minus the survival fraction, right? And it's just done it like this with a single parameter so that we can try and estimate what the cell kill is due to radiotherapy at the successive time points. So I irradiate, I kill off a certain proportion of cells, and then the dead material goes into this second compartment for necrotic dead material. So cell death, E to V, natural cell death, is a source term in here, and we have an additional source term due to radiotherapy. So every time I irradiate the tumor, I add to this necrotic pool, this pool of dead material. That dead material will itself decay and be degraded by immune response or whatever it might be at a certain rate zeta. Okay, does that make sense? Good. So that's our model, and again, it's really, I'm trying to keep this as simple as possible so that there's some hope that we might be able to fit it to the data. Yes? So again, you're right, it could be, and we could introduce, I mean, I'm not saying that this is a unique explanation of the data, it's one possible explanation. And indeed, it is possible if you think about immune responses, in certain cases that can be impaired. And I guess really, so this is just one. It could be sensitive cells. So if we wanted, we could have introduced a sensitive cell population, whatever. Yeah, so it's just one possible explanation. And I guess what you could do is have a series of candidate models that could explain the data, and then you design an experiment that enables you to distinguish between them in much the same way. But so we haven't done that, but those would be really nice and interesting things to do. So the way I would interpret this in terms of differences in clearance of dead material is how strong your immune response is, whether you're able to recruit macrophages into whether it's immune suppressed or not. So, and that's one possible explanation, but it is by no means the only one. So yeah, good points, really nice questions and comments. And so what, in truth, when we did fit this 2-compartment model to the data, so this was actually the results that, and so we have, I think, about 40 or odd different patients where we have this nice time-resolved data, and we are indeed able to fit them. And these are, I think this is maybe, typical patient, and you can see the confidence intervals, and so we can actually then predict, as you spotted with this pseudo progressor here, we can predict what's going to happen to them at the end of their course of treatment, where the tumour trajectory is headed. And you can see that in some of these cases there's going to be a rebound. In others, there's still they seem to be still decaying for some time, but we could predict when they might start to regrow and when you might want to restart therapy. So point being that the 2-compartment model does indeed provide a sort of unification of these different types of growth dynamics. And indeed, in fact, the way that we did this with our 44 patients is we did some approximate Bayesian computation to fit our populations. And this is just a summary of the fits of our population. And we've coloured the fits based on whether or not we judge them to be a fast responder, poor responder, etc., etc. And I guess the thing that the squares that I'd like to draw your attention to are those that have got boxes around them, which really the point being that this fit seems to sort of stratify or cluster the patients in terms of parameter space according to their outcomes, which I think sort of tells us something about subject, assuming that this model explains the biology, big assumption, but that it does seem to cluster the patients in parameter space. So let me see, I was looking at this morning and trying to make sure I understood it. So for our poor responders, we're seeing very slow rate, this is log scale, a slow rate of clearance of the dead material, which is what that's saying is that the volume that we're seeing may just be the accumulation of dead material rather than, say, the accumulation of radio resistance. So there is a hypothesis to test. Yes. Yes, so if you look at the top, log K, K is the carrying capacity. Yeah. Yes, yes, from the data. So we went through, we had 44 patients, we did ABC to look at the distributions for each different parameter group, and this is the results. And I think interesting to see how there does seem to be some separation between the different cell types. So again, a very simple model, designed to be at the level of the data. You can question the hypothesis, fine, I can question it myself, but it's really the principle that's important. And really the key here would be then sort of to have better data where you can test alternative hypotheses and explanations of the model fit. And indeed, so like you spotted on the previous slide, what we can do here, this is in terms of, I guess, if you know someone's going to be a poor responder, how quickly can you work that out from the data? So we can use the time course to try and predict how quickly we can classify patients. And then that would be useful because if they're not responding, don't keep giving this to them, do something else. And so this is one of the other values of using this sort of study. And so you can see it's sort of as you might expect. We've got different sort of, let's see, this is a good responder, plateau. I think this one must, oh yeah, sorry, this one's just a poor responder. This one plateaus, this one is, it progresses for a bit and then starts to respond. And you can see that our certainty in the outcome, how that increases over time. Some of these we're able to infer, I guess, some of them quite accurately at early times, but for others it's more challenging. So I guess really this is just to illustrate what you can do with these sorts of models. And I guess if you have a database of the different sorts of patients that you can use to inform for, say, following patients, then you can use that, I think, to improve the accuracy of your predictions and try and infer what particular class a patient is in and then respond accordingly.
Hi, I had a question.
Oh, yes,
Can we go back to the previous slide?
I can.
Yeah, can you talk about pseudo progression, the label, and you've made a box, and that is only present in RT cell kill log Y
That's there.
graph, the above one.
Pardon me?
Also, yeah, there's a small tinge of like,
And I guess
yeah,
up here,
yeah.
pseudo progression.
Yeah, can you explain? I would love if you could.
I think it's quite complex because with pseudo progression, I think what's happening with pseudo progression is you're seeing. So the reason why it seems to get bigger is because the dead material is not being cleared very fast at all, right? And so it takes time. So you irradiate, you grow more and more dead material. Eventually it starts to dissipate. As you grow more dead material, then the proportion of your tumor that comprises proliferating cells decreases. So your reduction in tumor volume when you irradiate gets smaller, but eventually you start to degrade that dead material. That's what I think is happening in this model with the pseudo progressors. I hope that makes sense.
I think what I understand is that instead of like measurement of tumor growth, it is measurement of increase of dead material.
Well, I mean, that's one thing, but it's also how quickly it gets cleared. So cells can be differently response sensitive to radiotherapy. They can also, the rate at which they clear their dead material can be different. The rate at which they proliferate can be different. The patients can be different in a whole host of different ways. So I think it's quite complex. So what I've just said to you is my interpretation of why someone is a pseudo progressor. So it's a combination thing. And you can see the heterogeneity in the patients. Not all patients, their tumor cells will proliferate at the same rate. Not all tumors will be equally sensitive to radiotherapy. And I guess you get a sense of that, I guess, from these fits. Yes. Yes. Well, I guess you can see that some of them are better than others. I mean, I think probably a nice thing to do would be use this data to kind of then generate a virtual patient cohort. That would probably be the sensible thing, to get guesstimates on realistic ranges for all of these parameters. So that would be probably, I think, with a sample size of 44, I think the fact that we could actually see segregation is quite good, right? So this is more illustrative than, I think, a panacea. Okay, thank you. La-di-da. So what have we done? So our initial very sort of naive model can't explain the clinical data. We've proposed a new model based on the hypothesis that irradiated cells are not cleared immediately. Under that hypothesis, we can fit the data and we can see that the patients cluster according to their inferred parameter values. And if you want to learn more, read the paper which appeared earlier this year. Okay, so what have we done so far, right? It feels like we've already covered a lot of ground. I'm feeling a bit weary, but never mind. So really the models that we've used have been fairly classical, simple ODE models, but I think the insight that's gained by how we use those models, how they interface with the data, I hope we've seen that that can generate new insight. I should stress that those data sets were very small and simple, very crude, but still, even so, when we combine them with the data, I think we can learn more than just simulating the model or just looking at the data alone. What happens when the data gets bigger, as it does? What can we do? and what other mathematical approaches can we, shall I keep going or should we have a break, actually? Shall I keep, yeah, so I'll keep going. Are we feeling strong? No? Okay. It will be a slight change of tack. Yeah, there is coffee at 11. You can almost smell it. So what about other mathematical approaches? Let's try something different. So, yeah, this was just to check how everyone was feeling. So we're good for another half an hour. I'm not going to do everything because I think that will be too much. So what we're going to do is do topological data analysis. That's what we're going to do. So it will be something different. So topological data analysis is a field that I never thought that I would be talking about topology. It wasn't a course that I did as an undergrad, but I think it's got a real lot of potential in terms of analysing big data. We can look at all of these images here. We can see that there's a loop. Our eyes are very good at identifying structure. If we look at this picture of Stonehenge, which is noisy, we can still infer a loop might have been there initially, right? So how can we formalize what our eyes see? How can we detect and quantify structure? And how can we apply that to big data? So I'll give you a sort of quick 101 on persistent homology, which is a branch of topological data analysis, just to give you the principles. And hopefully this will be okay. So you can see here, we've got a point cloud. These could be a bunch of, think of them as a bunch of cells, maybe a bunch of immune cells around a tumor nest, if you want, or just a bunch of points, whatever you like. And hopefully you can all see that it looks like that these points are on a loop, right? Give or take some noise in the data. How can we formalize that? How can we quantify what we see? So the way in which we do that, we put a ball around each of those points of radius epsilon, and we gradually make the balls bigger, increase the radius epsilon, and we see what happens, okay? So let's do that. And as you might guess, as the balls get bigger, eventually the radii, the discs from two nearby points will overlap. When that happens, I say that I connect those two dots and call them a single component. So what happens, as you can see here, these, or maybe clearer here, so these two dots, their discs overlap, and so I make them a single connected component. And the way in which we kind of summarize that is, as we start off, we have, I think, 20 different dots, 20 individual connected components. As I increase the size of my discs of radius epsilon, some of these discs will overlap. I will kill off one of my connected components because it's fused with another, and I will keep growing the other connected component. I will keep track of that. Does that make sense? And if we keep going, increasing the radius of our discs, then what can happen is we can see the formation of loops. So a loop is, there's a loop in here, so where we've got pairwise connections, our discs overlap pairwise, but there's a gap in the middle. We call that a loop. So you can see there's a very small loop that's formed just around and about here. It's in there. So we keep going. And then you can start to see the formation of this prominent loop, the one that our eyes can detect very clearly. It's forming here. Other connected components are fusing together, and so we see attrition in terms of the number of connected components here. Okay? And then we keep going. And then eventually we cover over, we have just the whole space is gone and our loop has disappeared. And so the barcodes that you see here are basically a multi-scale summary of our point cloud. So giving us information about the connectivity of points and also the structure, which in 2D we can think of as just loops in the data. And you can see that the length of these bars is giving us an idea of how long-lived or persistent they are, how prominent, whether those features are prominent in the data. So features which are very short are, or short-lived, we might ascribe to noise, for example. Does that make sense? Okay, so now you know about as much about topology as me. So let's keep going. And how can we use this? So this is just, I guess these concepts extend to higher dimensions. If you want to have a 3D or a higher dimensional, you just get higher order structures. So if we think in 3D, then you can form, rather than loops, you'll have voids at the center of a point cloud. So you can identify a higher order structures. So yeah, that's all great, Helen, but what can we do with that in biology? And so I think we'll probably just do this and then we might stop, is looking at tumor vascular networks. So how can we characterize the structure of tumor vascular networks? How do those change over time? How do they change following exposure to treatment? Treatment might be, yes. Yes, yeah, you do, but you do pairwise, you do it sequentially, so it's a multi-scale summary, and so you can see, it gives you an idea of how the structure is changing. So eventually, when my point cloud was all covered over, that was exactly the situation we got to, and you've just got one connected component, which is all of the dots have been joined together. So completely correct. It's probably not that meaningful. But I mean, it tells you that, I guess, by that length scale, when you've just got a single connected component, all the dots have been joined up, there's no more structure in the data. So it's giving you, if you like, an upper bound on the length scales of interest in your data. Well, you connect them. I mean, so you connect them based on a distance metric. And I guess here I'm using Euclidean distance. If you want, you could weight these in a whole host of different ways. There's all sorts of extensions that you can make to this. You might connect things that have different, you might have red dots and green dots. Do you want to just connect all the red dots? Do you want to connect red dots to green dots? What do you want to do? And you can have, you know, so there's so this is just the simple basics. But once you understand the basics, you can think about lots of extensions. And indeed, these are things that people, myself or my group included, are working on, because particularly when you look at sort of multiplex imaging data where you've got lots and lots of different cell types, how can you apply these sorts of methods? And that's really where we're sort of going. I won't get a chance, I don't think, today to talk about that, but that's the reason why we're doing this. And I guess what I'm going to showcase here is how we can apply these methods to vascular networks, but you can apply it quite broadly across a whole host of different things. Do you have, is that a hand or is that just? Well, that's the point, you don't. It's a multi-scale summary. So this is the beauty of persistent homology. Because the length scales emerge, you can see them from the barcodes, right? So you don't have to prescribe your length scale. Yeah, you see when features appear and when they disappear, what length scales, and that, I think, is the power of it. Whereas many say statistical methods will kind of fix a length scale, right? So that's one of, I think, the advantages of these sorts of methods. Yeah. Right, so what I'm going to show you is some work that was done in 3D vascular networks. And this was done quite a few years ago now, which was very much, so this is probably started maybe 2018, 19. And at that stage, the algorithms were just coming out. They were full of bugs and it was awful, awful. Nowadays, there's lots more people doing this. You can sort of, you know, there are sort of algorithms available, you can download, and you can use them quite quickly.
Do they work well in, say, dimensions of as large as, say, 7,000, 8,000 dimensions, or it's still low dimensional?
I think probably low dimensional. I mean, yeah, I mean, it's still new stuff. Yeah. Yes. Yeah. 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 In principle, I mean, yeah, you might be better off. I mean, so some of the other approaches that you can use, so it's not a cure-all, right? It won't work for all data sets. I mean, so again, won't have time to talk about today, but some of the things, if you think about it, you could analyze using spatial statistics, you can use topological data analysis, you can also use methods from network science. All of these are nice methods to use. The question is, which is the right one for my question? So all I'm doing today is showing you a method and showing you what it can do, but it doesn't mean it is the method. And indeed, I think there is a question when you're analyzing data about trying several different methods. Are your findings robust across those methods? If they are, great. If they're not, then what does that mean? Ask a question. So this is just one new method to add to your toolkit. Okay, we'll keep going. Yeah. So I guess I've sort of said this already. So in contrast, you can analyze vascular networks using statistical metrics. People have done this. You can look at branch lengths, numbers of bifurcation points per unit length, et cetera, et cetera. And these sort of statistics can and have been very useful and informative. However, as was mentioned, they focus on a single spatial scale. You choose your length scale, whereas what we want to do, and they don't say anything really about the structure, the presence of loops or higher order structures, voids, that we might see in our data. So for example, in a vascular network, you may have an avascular region in your tissue, which might be sort of a tumor nest with a central necrotic core. So it's very difficult to pull out those features, and it's very difficult for the eye to do that as well. And it's very difficult to do that well, it's difficult for me to do it in 2D, let alone in 3D. So the nice thing is that these methods can do that. They've got, much better eyesight than me. So what we did, we've got data from 2 different types of microscopy, or we've got one which is live cell imaging using a window chamber assay, so you can view the same tumor over different time points. We've also got ultramicroscopy data where the tumors were excised, and then we could section across across the entire tumor. And the tumors were either control untreated, or they were exposed to vascular targeting agents, or the tumors were irradiated. And what we wanted to do is look at these sort of time courses and see if we can detect any changes in the vascular architecture using pretty much the methods, or very close to the methods that I've just described. And just to give you more detail, so credit to Bernadette, who was the person who, as part of her PhD and postdoc, enjoyed grappling with the algorithms, and it was awful, let me just say. So, and her data is huge 3D data sets of vascular networks, and I mean, I think tens of thousands of vessels in here. So huge, huge data sets. And we spent a lot of time thinking about how to apply TDA to this data. And the method that we came up with, which was to put, if you like, a dot at the center of our vascular network, and then we inflate a ball from the center. Prior to that, so these data with whatever 30,000 or more nodes, etc., is too big for the algorithm. So what we had to do instead was subsample it and then apply our TDA and do that several times over, because just couldn't compute, right? Took days to do this. So what we do is subsample our vascular network. We put a seed at the center and gradually inflate this ball in 3D. And then as the ball expands, we see which subsample points are inside the ball and which ones are outside. The ones that are inside we can connect in the way that I've just described. And from this, we can build our topological summaries in terms of connected components, loops, and also because we're in 3D, we also get voids, yes. So you can't really see it because it's small on here, but within that, that's been segmented. So somebody else's thesis is actually doing the segmentation to pull this out, right? So we've got the X, Y, Z coordinates of all the nodes, et cetera, and to make sure that that all makes sense. Yes, yeah, and then you know which ones are connected to which. So all of that information is encoded, right? So you will have the coordinates. I guess they may have made some, you can probably do piecewise linear. Okay, so, but anyway, you could, so as I said, this was somebody's thesis doing this, just doing the segmentation. It was, again, awful. But they did it. And so we took that data and we apply this filtration, we call it a radial filtration because we're just inflating a ball. And then from that, we can look at connected components, loops, and voids. And with fairly, and I guess sort of short-lived barcode connected components, if you imagine as this ball increases, short-lived connected components would be kind of, like you were saying, vessels that we've subsampled that are wiggling sort of, if you imagine, I can't, maybe I'll draw it on the board if I can. So if you have, so that's where I've got to. If I have a vessel which is going like that, say, then very short-lived connected components you can think of as giving some sort of idea of either noise or tortuosity. You can interpret it in that way. So what does it look like? So perhaps in the interest of time, let's just look at some of these results. Maybe let's look at loops down here. So the orange bars are showing you how over time across my tumor as a whole, the number of loops changes. And for my untreated tumor, unsurprisingly, as it's getting bigger, we can see an increase in the number of loops in my vasculature. And this is over, I think we had about 5 or so tumors to look at. And then we've got 2 different treatment groups. So one, DC 101, I think this is inhibiting angiogenesis, inhibiting the formation of new loops. And then, as expected, we see a sort of reduction in the number of loops. So that confirms that it is indeed working as it should do. This, if that says D44, this is a vascular promoting agent, and indeed we're seeing an increase in the number of loops. So what this what this analysis did is confirm that these drugs are indeed working in the way that we expect them to do. The pie charts on the right-hand side is really just giving you, because we've got sort of 3D spatially resolved data, we can do a sort of, I guess, smaller volume, sub-volume analysis. And so what this is showing you how over time, at different depths from the tumor center, so tumor center, this is the outer boundary, you can look at the number of loops here, 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 and how those change over time. So it's giving us spatially resolved information about how the structure of the vascular network is changing. Okay. And if you, I think it's all in the paper if you're interested, but really just to show you what sort of information can be pulled out. And I guess what you can see here is when we're giving, I guess, this anti-DLL4, which is promoting angiogenesis, you can see how you're getting a very strong increase in the numbers of loops just inside and on the tumor boundary. So it's quite localized. And so you get much more, you can get quite detailed information about where the angiogenesis or vascular remodeling is happening. We also repeated the same analysis, but when the tumours were irradiated, either a single large dose of irradiation at day 1 or smaller doses over a period of 5 days. And what we found here, so this was interesting because no one's ever really been able to quantify the changes in the vasculature following exposure to radiotherapy. And so this enables us to do that over time. So you can see in the controls, we've given, you can see in the controls, so they've not been irradiated, you can see how the vasculature is remodeling over time. If we give, let me get this the right way round, if we do a single dose, large dose of radiotherapy, we see, I guess, initially we've killed off a lot of the vessels, and then we see, I think, a fairly marked, very strong angiogenic response, which may be due to all of those dead cells stimulating a very strong immune response, and those immune cells acting as pro-angiogenic and recruiting, producing angiogenic factors to stimulate angiogenesis of the vessels. But I guess, and then if we do fractionated radiotherapy where we're hitting the tumour daily, we can see a stunted response that the new vessel growth is definitely being inhibited here. Okay, so that's really just to show you what you can do and the insight that you can gain from using topological data. And I think we won't go there. Right, okay. I have 10 minutes. Yes? Yes, yes, indeed. And I guess the point being, you can only keep your mouse under a camera for a certain amount of time. They've got to be anaesthetized, and so you have to be, there are considerations as to how long, but certainly other collaborators that we have at the moment, that's exactly what we're looking at. Because again, where that goes to, and this is something else we've been thinking about, is linking the form and the function. So now we can see the vasculature. The question is, to what extent do the number of loops have anything to do with the tissue oxygenation? And I imagine will know that tumor vasculature is very immature. It's not very high functioning. So there's lots of redundant vessels. And so there's a question about the extent to which the numbers of loops, what does that tell us about the actual oxygenation that's being delivered to the tissue? But indeed, that sort of analysis is possible. And some of the work we've just had a paper published in PLOS Comp Biol where we sort of starting to look at that, if you're interested. So just in this final bit, if that's okay, if you've still got the energy, is really what we're going to do here is try and show how we can use the same sort of methods, but combine them with a model of angiogenesis. And this is really going back to like with the clinical model on radiotherapy, where I've got a mechanistic model and I'm going to use TDA to characterize. I'm going to use my ABN to generate, I'm going to use it to generate synthetic vascular networks. I'm going to analyze those using TDA, and I'm going to see whether or not there's any relationship between the parameters that we use to generate the networks and if I classify them based on their network structure. And this is really just to give us some sort of insight as to whether we might be able to learn something about how the parameters driving the generation of networks in a particular tumor, right? So that's the idea. And to do this, we decided to pull off the shelf probably a sort of classical model of angiogenesis due to Mark Chaplin and Sandy Anderson from the last century. Very simple agent-based model where we have a source of angiogenic factors on the right-hand side. That could be our tumor, if you like, and that diffuses through the tissue. When it reaches blood vessels, it will stimulate those endothelial cells to form sprouts, which migrate by chemotaxis towards the source of chemoattractant. Sprouts may fuse together to form a loop, So you can see the different things that can happen here. So two tips might fuse together and anastomose. A tip might fuse with an existing sprout or vessel. And so you can have tip to sprout and tip to tip anastomosis. Equally, a capillary tip sprout that's migrating out might bifurcate and branch into two new sprouts. And all of these rules are encoded in our model, or Mark and Sandy's model, should I say, of angiogenesis. And we use this to generate synthetic vascular networks. So in essence, the tip cells are performing a biased random walk on a regular lattice in response to a chemoattractant, but we also have a haptotactic gradient across the tissue, right? So there's going to be two key parameters of interest. So these are the sorts of models that you can generate, or the sorts of networks. We can see stunted growth, we can see a little bit greater growth, you can see these very dense hairballs of vessels forming, and you can see very rapid growth with little anastomosis. So as we vary system parameters, we can get this sort of zoo of different types of networks being generated. And I should say that in the synthetic networks that we generated, we focused on varying just two parameters, so endothelial sensitivity to chemotaxis and also to haptotaxis. So I'm just varying those two parameters to see what sort of networks we can generate. And these are illustrative of that. So then the question is, how do we quantify those networks? Because what we wanted to do here was to, if you like, benchmark our TDA metrics with more routine standard statistical descriptors. Because if the standard statistical descriptors can do the job, you don't need TDA, right? So what we did was characterize things like how many segments we have, how many tips have we got, what's the penetration depth of my vascular network. And these are all sort of routine things that people have used to characterize vascular networks in the past. And if you look at just some typical networks, this just gives you an idea of what those sort of quantities, how they change as my network progresses, as it grows from, say, left to right. And it all happens in a fairly obvious sort of a way. And so we've got a chemotaxis-dominated haptotaxis and chemotaxis plus haptotaxis simulation. And the results down here just summarize how those different sort of numbers of tips, segments, and penetration depth, how those change over time for these different simulations, just to give you an idea of what happens. Going towards topological descriptors, then the sorts of things we can keep count of. So beta 0 is just keeping count of how many connected components I have. Beta 1 is how many loops. So that's really Betty numbers if anyone does topology. So you can see here I've got two connected components and no loops. Here I've got one loop, and so on and so forth. So you can count, you can add to your arsenal of standard statistical descriptors to topological ones, and these are my, say, beta 0 and beta 1 as a summary, a topological summary of my vascular network and how it's changing over time. There is a question about when we apply our topological data analysis to our vascular networks, how should we do it? So with the biological example on the vascular networks in 3D, remember we put a point at the center of the network and we inflated a balloon. Now I've got a 2D synthetic network. How should what filtration should I do? How should I generate my multi-scale summary of the data, right? And this is where it starts to become a bit of an art, if you like, right? There's different ways you can do this, and let me just talk you through a couple of those that we tried, because it's not obvious what's the right way to do. It's a bit like if you're doing asymptotics, you might not know straight away what's how should you, where should you put your boundary layers, what scaling should you use, etc. To me, I think a challenge is knowing what sort of filtration to use to apply to your data. Yeah.
First example that you started with points on every dot, and then you do those.
Yes, but that's.
Does that also matter how many you start with?
How many dots?
Yeah, and then because in this.
No, so I think the filtration is how you connect the dots. It's the method that you use. So in that case, it wouldn't matter if I had 100, how many, the method could still be applied. That doesn't depend. I mean, how computationally efficient it is might matter, which is why when we did the 3D vascular networks, we subsampled the data, right? So the filtration, which is how you kind of connect the dots, that varies. So in the first case, I put a ball on each point and expanded it. I could do that with 100 dots or whatever I like. I could have done that with my vascular networks, but it wasn't very informative. So instead, we did this expanding ball thing. Here I'm analysing 2D networks, and there are similar questions as to what's the best way to generate my multi-scale summary, right, or to do my filtration. And so one way to go is, now let me get my brain back into gear, is what we're going to do is we're going to sweep across my network from left to right. So what I've done here is here's a binary image from a simulation. So the red dots is where I've got vessels. And I put a point in every cell that contains a vessel. And then what I'm going to do is I'm going to sweep a plane from left to right. And at a given point, it's where this horizontal blue line, vertical, yeah, I need coffee, where this vertical line is. And so I look at the points to the left of the blue line. So at this time point, I've got these points. So these three, they form a connected component. I fill it in because they overlap, right? So there's one single connected component. It also connects there. So I've got one connected component here. And then I've got a second connected component with these dots, and then a third one down there, right? I move my vertical line further across. I bring more points into the domain, and then I grow my filtration, right? And so this is what we generate if we do this left to right sweeping plane filtration across my data to see where are the connected components. Here you can see, this is quite a simple example. I'm picking up this loop, which you can see here in the data, right? Does that make sense? Right, I'm gonna sort of go to the, we're sort of should be finishing now. Sorry, this is taking longer than la-di-da-di-da. You can do all of this, and in fact, you can sweep left to right, right to left, top to bottom, bottom to top, all of these different things we've tried, and these sort of are just summarizing, as we do that sweep across the networks, how many connected components, how many loops, as we go in different directions. And you can do this across your different images, different networks. And then, so just to summarize what the method is, so what we did was generated a bunch of synthetic networks by varying chemotactic and haptotactic sensitivity. That gave us a bunch of different networks, but we know what parameters we used to generate them. We analyze those networks using either, there, that wants me to stop. I'll get to the end of this and then we are going to stop very quickly. Standard descriptor vectors, tips, vessel segments, etc., but are supplemented by our topological ones. Then what we did was we looked at different combinations of those features from our data, and we used k-means clustering to cluster the data according to the descriptors. And sort of long story short, what we found was that no single descriptor vector adequately segmented the data into clusters. what we ended up doing was taking pairs of and the topological ones outperformed the standard ones. And what we found is if we took double descriptors, so that might be left to right, right to left filtrations, that those produced the most meaningful clustering of the data, as you can see. So these are just these are just different combinations of pairs of descriptors to show you how, and different colors are different clusters. So we don't like this one because it clusters these networks with these. The axes here are the parameters that we use to generate the data, right? So if you look at this clustering, that doesn't look too bad. It's separating these networks into one group, these into another, et cetera, et cetera. So what we find is if we use the topological descriptor vectors are better at clustering the data. We use combinations of those topological descriptors to get what look like meaningful sort of clustering of the data in terms of the parameter space. And again, this just is to sort of give you an idea that those networks that were generated are close in principal component. We sort of projected this down onto the first two principal components. We used our elbow method to work out how many clusters was appropriate. Okay, I think given that we are well over time, let's just la-di-da, you can apply it to other methods. I think, yeah, I won't bother. There was another bit of stuff in here that I'm not going to go into where we've applied these methods to data from lung cancer. If anyone's interested, happy to tell you about that later, and a variety of other things. So what we've done this morning, we've covered a lot of ground. Thank you for your patience and for your questions, and really just walking through how we can link mathematical methods with experimental data, and sort of showing how the methods that we use as the data changes in its quality and quantity, how different methods can be useful. And I think I will stop there, and happy to take any questions, but maybe you might want a break first. Thank you.